Hello, good morning, everybody. It's my pleasure to welcome you to another webinar of our Bioinformatic series. So today's speaker will be Adam Seglam. You already know him from the previous webinars that he gave in this series. And the topic of today's webinar is the downstream analysis of single-cell RNA-seq data using the standard procedures. So what we will talk about in this webinar today is we will talk about the gene enrichment analysis. What is it? We will also talk about the cell pseudotime trajectory. We will discuss what the RNA velocity is and what assumptions it is based on. If you have any questions during the webinar, please feel free to write them in a chat or raise hand, and we will try to answer all the questions that you might have in the end of the webinar. So please, Adam, you can share your presentation now.
Thank you, Katarina, for the introduction. Okay. So welcome to this webinar series. This is the fourth one about the trajectory inference in single cell analysis. So today I'm going to talk about what trajectory inference is and when it is useful. I'm also going to mention some other dimensionality reduction methods because some other methods than discussed in the past webinars are used in the trajectory inference tools that I'm going to present also today. Then I'm going to talk particularly Monocle and RNA velocity as the trajectory inference or trajectory analysis tools. So we will start with the trajectory inference, an introduction to the trajectory inference. What do we mean by trajectory inference or trajectory analysis? So imagine that you have this UMAP that you also have seen in the previous webinar series. So each dot would represent one cell, and they are already labeled here, as you see. Do pay attention to the rows for now. So the colors represent the different clusters or different cell types. So you have a cell somewhere here, and it has a certain transcriptome profile. Another cell next to it will have a similar transcriptome profile, but still slightly different. So that's why they are apart. Otherwise, they would be superimposed. And another cell that is further away from the second one has, again, a slightly different transcriptome profile. But these slight changes in the transcriptome profile are, of course, due to some biological variability, but also due to the fact that not all cells at that time of the experiment were at the same status. It's like some cells were already dividing, some cells have already divided, and some cells, they are just in the middle of the cell division, for example. Or if you think from a stem cell development perspective, you might have initially a stem cell that start to differentiate, but in that tissue, you will have different cells that are at different stages of differentiation. So here you have all these mixtures, and therefore, the cells that are at different differentiation stages will be close to each other depending on how close the differentiation stages are. So we can see the trajectory inference as a way to measure the fate of a particular cell. So I start with a cell located here, and knowing that the other cells at other stages are located here, then I can guess, or I can assume that this cell will probably evolve along this trajectory. So this is what we mean by trajectory inference or trajectory analysis. So in this case, it's a pancreas tissue with all the different cell types in the pancreas. And the arrows indicate the direction that the cell would follow if they had to differentiate. So this is determined, of course, bioinformatically. So there are different types of trajectories. And therefore, there are different tools of trajectory analysis. So by the way, I use the word trajectory inference and trajectory analysis as synonyms. Okay, so here you have, for example, a cyclic trajectory that you would have when you have a cell cycle, for example. So a cell would start somewhere here, or there is no real starting position. So it would be anywhere at one of those vertices, and then move in that direction, then there, and then come back. So this is a typical cell cycle or cell division trajectory. Then you have other situations where you have a linear trajectory. So you have a stem cell at the beginning, and then it fully differentiates into a particular cell type. So there is no way back. It's purely linear. Then you have bifurcation trajectories, like you have again stem cells, and this cell can differentiate into several cell types. So this would be convenient for that case, etc., etc. Then you have more complicated trajectories on the right side. So yeah, this you can really read the paper from Salens published in 2019. They really give a very good overview on all the different trajectory analysis methods. So again, this is from the same paper. So you have initially your single cell expression count matrix, and then depending on the type of trajectory analysis that you would like to do, whether you have a cell cycle, whether you have bifurcation, branches, etc., then you would use one of those methods here. Okay, so maybe some of them might be familiar. So I'm going to talk about Monocle today for sure. And the others, they are also available so that you know it. So they use different tools, different approaches, like different dimensionality reduction methods. So we have already talked about PCA in our previous webinars, TSNE. But for example, Monocle, if you follow this blue path, it uses ICA, independent component analysis. So I'm going also to talk about that. Yeah, so the different tools will use different dimensionality reductions, and then after reducing the dimensionality, it will use different trajectory modeling algorithms. Yeah, again, in the same paper, you will see a very good extensive list of different tools that can be used for doing trajectory analysis, whether they are Python-based or R-based. Like, I'm also going to talk about RNA velocity or scVelo. This one is Python-based and Monocle is R-based. And you can see in which cases you can use the different tools. For example, PAGA would be useful for if you expect to have a cyclic trajectory or linear trajectory or bifurcation, etc. But some other tools, they are not really appropriate for that. For example, Slingshot, which is also a widely used tool in trajectory analysis, it cannot detect any cyclic trajectory in the data. Yeah, so I'm going to talk about some other dimensionality reduction methods. As I said, they are used in some of those trajectory analysis tools. So that's why I thought it might be worthwhile to talk about them. So you remember that PCA, if you have this kind of data, like, so each dot represents a cell in 2D. So imagine you have gene 1 and gene 2, gene 1 on x-axis and gene 2 on the y-axis. And you have a kind of distribution that looks like an X. It's obvious to our eyes. But if you use PCA, PCA will basically find the directions of highest variance or highest variability. So the first component is the one that would that indicates the direction of the highest variance, and the second component will point to the direction of the second highest variance. In this case, since our data is 2D, there are only two components. But if you had more components, then you would more dimensions, more genes, then you would have more principal components. And at the end, we would still keep two for visualization purposes. Yeah, so PCA indicates the directions of highest variance. And the weight of these directions indicates the the variance in that direction. So the second component will always be smaller than the first in PCA. In ICA, however, which is independent component analysis, the idea is to find the independent components that when you sum up their effect, it would be possible to explain the data. So here, the directions are basically pointing along the branches of the X shape. Okay, so you have the first component pointing along one branch of the X and the other one pointing along the other branch of the X. And in this case, the weight or the size of the vectors that point toward these directions, they are more or less, they have more or less the same amplitude. So there is no difference, no big difference among the weights of these components. So here the components are assumed to be independent. That's why it is called independent component analysis. So maybe to explain a bit further, so ICA has been used to reconstruct sound. So imagine that you have three people talking in a room or three different instruments being played in a room. So you would see these three curves, three instruments, and then you have three microphones located in that room that would measure the sound level, the sound signal coming from these three instruments. So because you have three microphones, you have three curves. So you detect three curves. And from these measurements, you would like to reconstruct the signal that would come from each instrument individually. So this is the true sources. Each of these instruments would have this kind of signal. Okay, it's just a simulation. So let's imagine that you have instrument one that is red, instrument two that is this yellow curve, and instrument three that is this blue square-shaped curve. And this is the true source. So the mixture of these three signals will give any of these three output detected by each of the microphone. And if you use ICA on this original data, then it will basically reconstruct the sources like this. So you can see that the yellow curve here, the square-shaped curve, seems to fit very well the blue curve here. By the way, the colors among the figures, they don't match at all. They don't mean anything. Okay, so don't think that the red curve in the second figure represents the red curve in the first. They are totally independent, because here on the second plot, we talk about the individual sources, while on the first plot, it's a mixture of these three, a random mixture of these three detected by each of the microphone. So when you use ICA on this original three signals, you can detect the three sources. But here there's just one inaccuracy, which is the red curve, the sawtooth curve that was expected to match the yellow one here, but as you see, it is just the opposite that has been found. So the method, of course, it has some limitation, but as you see, it can detect also very nicely some of them. And if you had used a PCA on this original data, then you would have detected this, which has nothing to do with the sources, the true sources, as you see. So if we do the analogy at single cell level, you can think of these three sources as the raw gene expressions. The true sources would be the true biological sources. Okay, so the ICA basically tries to capture the biological reason of the change in the gene, in the raw gene expression. This is what it tries to do. So if we compare ICA to PCA, both are linear methods. First of all, ICA estimates the sources that compose the data, while PCA finds the direction or the components of highest variability. In ICA, sources are assumed to be independent of each other, and sources are of equal importance, while in PCA, the components are sorted by variance, so they are not equal. Another method for dimensionality reduction is the diffusion map method. So imagine again you have 2D data where each dot represents again one cell, let's say on gene 1 and gene 2. So gene 1 would be on the x-axis, gene 2 on the y-axis, and the cells are distributed like this depending on their gene expression. So you see that here, if you use ICA or PCA, you will definitely not be able to capture this obviously path that might indicate some biology over there. So we see this by eye, that it has a spiral shape. So there is probably something happening along this direction, but ICA and PCA would not be able to capture that because they are linear methods. They just find the components along straight lines. So in diffusion map, we first calculate the distances between all pairs of points. So you will get something like this. Of course, not all distances or connections are plotted, otherwise the figure would not be readable. But yeah, you can imagine that from one point here, you draw lines to all the other points, and then you measure these distances. From the next point, you do the same. So you have a big matrix. We call it distance matrix. After you obtain that distance matrix, you apply a kernel that basically sets a threshold on the distance, like the distances that are shorter than a certain threshold are considered to have high affinity, while the distances that are longer than a certain threshold, they are considered to have a very low affinity or low probability. So basically, if you imagine a cell here, what is the probability of this cell going through this direction or this direction compared to going from here to there? Obviously, the cell has much higher probability to progress in the transcriptomic space, to progress, to differentiate into a direction of its own neighborhood, right? So that means that it makes sense to keep only distances that are shorter than a certain threshold. And when you apply this kernel to this data, then you basically get rid of all these long connections. So you have only the local connections. You have the local connectivities only. Once you have this graph, then you can easily detect the first diffusion map component that basically goes along this path like this, so which is plotted here. So as you see, it is a nonlinear curve. You will not be able to detect it with PCA or ICA. So you need a nonlinear method. So diffusion map is a nonlinear method that detects this component. So this would be the component 1. The component 2 would be the line that goes sideways. Okay, so then using these new components, so in diffusion map or the new components, if you use ICA, you can reduce the dimensions to then go for the next step that I will explain on the next slide. So the diffusion map, if we summarize, so it's a nonlinear method. The distances between points measure the probability to go from one point to another. So we use the principle or the assumption that a cell, when it evolves, it would evolve to its first a closer neighborhood than to another transcriptomic stage that is much farther away. So once we have reduced our dimensions, so imagine that this is our data in reduced dimension, we would like to detect the path that a cell would follow. So here we have a certain number of points, I think 10, and they are connected with each other. If you use ICA, then each of these numbers would indicate the distance between each pair of points. Or if you use diffusion map, then these numbers would indicate the probability of transitioning from one cell to the other. Yeah, so in ICA, the distance would be the Euclidean distance, but I think you can also use cosine distance, but then this would be used by another method. So once we have this set of points, the idea here is to find the branches that would give the minimum sum of all these distances. So if you, for example, here, the minimum, this is called the minimum spanning tree. So if you imagine that you change this connection, for example, this one here to this one, then the distance that was initially 8 would increase to 9, and this would increase the overall distance, sum of the distances, by 1 unit. So you see that this plain bold tree is the one that minimizes the sum of the distances. And this indicates a path, an estimation of the path that a cell would take if it had to differentiate. So if the cell starts here, it would go there, then there, then there, then follow this path, basically. So you see there is a branch here, there are 2 possibilities, and you have again a branch here with 2 possibilities. So this is the idea of a minimum spanning tree. In ICA space, the distances would be the Euclidean distances, as I said, and in diffusion map, if you reduce the dimension with diffusion map, then it would be the probabilities that would indicate the value of these connections. And by definition, the minimum spanning tree cannot have any cycle. It is impossible to come back to a point here because we minimize the distances between them, choosing the connections that have the minimum sum of distances overall. Okay, so then how is this used in Monocle? So Monocle, it's a tool in R that can do trajectory analysis. So initially you have your cells in a high dimensional space. Here it's shown in 3D. Then you do your dimensionality reduction with ICA. So you get this. All these cells are projected into this space in 2D. And then you calculate the minimum spanning tree with MST. So you get this. And then you can, once you have this tree, you can order the cells along that tree. So here you need to mention where the starting point is. So this method cannot tell you the start and end positions. Like if you have a tissue where you have stem cells that differentiate into a fully differentiated, fully mature cell, it will not be able to tell you that this point is where it should start or this point is where it should start. You have to help. So if you know that the stem cells are located here, then you would say this, it's the starting position. Then this would be the end position as it is the main branch. And the starting point is assigned a value of 0. And the end one is assigned a value of 1. So basically along this trajectory, you start from 0, you end at 1. And this trajectory is basically a measure of time, but it is not a real time. That's why it is called pseudotime. So that means that if a cell starting at pseudotime 0, if you let the system evolve, if you could do that, because your experiment is one snapshot, you have taken the transcript, the mRNA, and that's it. But if you could let the cell evolve, then it would very likely evolve in that direction at a later time point. And here you have a bifurcation indicating that it can also go in that direction. And on the right, you see some case with real data. So you need to specify the beginning, and then it recognizes the end, and then it tells you, it puts the cells along this trajectory. And you can plot the gene expression along that trajectory to see how it evolves along the pseudotime or along the trajectory. And lastly, I would like to talk about RNA velocity. So this method is based on counting the number of spliced and unspliced reads So what does it mean? Spliced reads are basically reads that are aligned purely to exons, like this one here. Or reads that align to exons, again, purely, but initially, you know, when you have mRNA, the immature mRNA has introns. And once it is spliced, this intron is gone, and you are left with this part and this part together when you do the sequencing. So you have this and this as one read. But when you do the alignment, you can find that this portion aligns to this exon and this portion aligns to this exon. So because they are fully aligning to exons only, those are considered as spliced reads. Unspliced reads are reads that contain some part of the introns, or they align only to the introns. Statistically, we have like between 70 or 80% of spliced reads and between 20-30% of unspliced reads. What we can detect with the different single-cell RNA technologies. So once we have the different counts for these, we can then use a model to try to estimate some parameters. So you know that mRNA is obtained from transcription of a particular gene with a certain probability alpha, or a rate of transcription alpha that we don't know, and it is different for each gene. But we assume it to be constant along the time. So it's only gene dependent. Unspliced mRNA is obtained afterwards, after transcription. Then the number of unspliced mRNA is described by U. You have then splicing with a certain rate, but here the rate beta is considered to be 1. It is just an assumption of the model. There are other models where they don't fix beta. And then you obtain, after the splicing, you obtain a spliced mRNA. And the amount of spliced mRNA is shown with S. And then once you have the spliced mRNA, the mRNA gets degraded with a certain rate of gamma. So if you remember the chemistry course that we had a long time ago for me, you can write the rate of the unspliced mRNA, the rate of change of spliced mRNA. So basically it is equal to the production minus destruction, or production minus loss. So you have transcription, so you get unspliced mRNA with a rate of alpha here. Also, you have a T, so which means that it changes with time, but actually in this model it doesn't change with time. It's just assumed to be constant. It's only gene dependent, where K is the parameter for the indicating that it corresponds to the K gene. Okay, so you have these equations, and if you solve them, you would get things like that. So the transcription is assumed to be on-off. So it either happens or not with a certain rate. And then you get that the unspliced mRNA at the beginning is 0. And once you have transcription, it increases. And as soon as you have some unspliced mRNA, you also have some spliced mRNA with a certain delay. So here you see that it starts increasing fast, but here S, it increases slowly. So there is a delay actually between U and S. And then they reach a certain steady state point, because the rate of generating unspliced RNA becomes equal to the rate of degrading spliced RNA, which is at this point. And then after that, once the transcription is off again, they both start to decrease. So this is the general solution of these equations. And the idea of the method here is to find the parameters alpha and gamma for each gene. I mean, gamma is general for all the genes, but alpha is different for each gene. So once you find alpha, then you can know how fast a gene is transcribed. And then this will give you an idea on the RNA velocity, basically. This is where the name comes from. And so in this paper from Lamano, published in 2018 in Nature, they measured the circadian genes. So you have, each row represents one gene, circadian genes. On the left side, the first column represents the spliced counts, and the right column represents the U counts, unspliced counts. So at time zero, whatever the time is, it doesn't really matter, we have a certain amount of spliced, if we just look at the top part, a certain amount of spliced counts for these genes, but slightly higher unspliced counts. Then 3 hours later, we see that they become equal. And again, 3 hours later, we have spliced counts that are more abundant than unspliced. And again, 3 hours later, the spliced counts start to decrease, like the unspliced ones, etc., etc. And when you measure this cycle, you see that it's almost 24 hours. Yeah, I mean, the 24 hours are there, basically, if you had another graph showing it. So you see that the spliced and unspliced counts in the circadian genes, they vary with a cycle of 24 hours, which means that one can use these information, spliced and unspliced counts, to measure the stage at which a particular cell is. So I told you, I think 2 slides before, that there is a delay between the curve of the unspliced and spliced mRNA. So here you see it for different genes, that the unspliced is always ahead of the spliced, which makes sense because you should first have unspliced mRNA and then have spliced mRNA. So you have this for all the genes here. Okay, so this is an example of pancreas data that I also showed on the first slide. So if you just focus on a particular gene, CPE, you plot its unspliced and spliced counts for all the cells in the data, all the cells. So unspliced versus spliced for the particular cell for this gene, and then for another cell, and then for another cell, basically all the cells in the data. So then you see this kind of curve that indicates you how the unspliced changes, spliced counts changes versus spliced counts. And the shape of this curve depends on the parameter alpha and gamma, the unknowns. So you fit that curve coming from those differential equations that I had mentioned previously, then you can determine alpha for each gene. And basically, just to explain it with basic words, the more this curve is away of this straight line, the higher the RNA velocity is for that particular gene. So you see, for this gene, the curve is very far from this line, then the RNA velocity is definitely high in this region. But for another gene, like INS2, you see that the curve is very close to the straight line, and the velocity is not so high. RNA velocity is not so high. And by using this information for all the genes, you have a velocity profile for a particular cell. And you can use this velocity profile for a particular cell and in its neighborhood to calculate the direction in which the overall velocity goes. So in this case, you get the arrows. And this arrows indicates you in which direction that cell would evolve if it had to evolve It doesn't have to evolve, but if it had to evolve, if it had to differentiate, it would evolve in that direction. This is what the technique helps you to infer. And the advantage of this technique, RNA velocity, compared to Monocle, is that here you don't have to mention a starting position because it appears already by itself, right? So it finds the direction of the arrows, and then you know that the start point is where the arrow is starting, and the end point is basically on the right side here, right side, alpha beta cells. Okay, so we came to the summary of the talk. So trajectory analysis in single-cell RNA-seq data is estimating the fate of a particular cell. There are several methods for trajectory analysis, and I have talked about two only. So Monocle, which is graph-based, and scVelo is the name of the tool that uses the RNA velocity method. And lastly, the choice of the appropriate method that you would like to use for trajectory inference depends on what you expect, what biology you expect in your case. So if you expect to have cell cycles, then you should not use Monocle because you only have branches with Monocle. Okay, but you can use RNA velocity. Or if you expect to have branches only, then Monocle would be suitable, as well as other methods. Okay, so that was it. Thank you very much for listening, and I'm happy to take questions.
Thank you very much, Adam, for a nice talk. So we have a couple of questions from the audience. I will start with a question related to the trajectory analysis. So the question is whether the bifurcation of cells in the pseudotime has the same probability for all the cells, or whether some cells are favored to go one path over the other.
So the bifurcation, it has the same probability for all the cells that reach that bifurcation point, yes. Okay. Because until that point, all the cells are assumed to be similar. It's only after they pass that point that they start to change.
All right, I hope that answers the question. If not, feel free to raise the hand and you can ask the question directly to Adam. The next question that we have from the audience is that also question related to the trajectory analysis. So basically when we have in the pseudotime the hours, like for instance 18 and 21 hours, whether this is a pseudotime or whether it's longitudinal related to the basically different collection points from the longitudinal studies which were collected at different intervals from the patients. Could you explain that, please?
Yes, so if the person means this, so here, yes, it is a true time here, but this is just to validate the method. However, if I may come back further, what did I pass? I think I passed it. Yeah, here, for example, 0.48 doesn't represent any time. It is just the value that you get along the pseudotime. All right, so it is not a real time, because when you do trajectory inference, you can never know the exact time to which it corresponds to. So that's why it's always normalized from between 0 and 1, 0 being the start and 1 being the end. But you cannot know the exact length in terms of hours.
I think Mohammed has a direct question that he would like to ask you. So I think, Mohammed, you are free to talk.
Yes.
Hello, do you hear me?
Yes, we can hear you.
Hello.
Oh, okay. Yeah. Thank you so much for this webinar. It's really very rich in information. And I have attended your first lecture. when you explained in detail how the single-cell RNA sequencing wet lab protocol is being done in the wet lab. And I remember you had a picture of a device where cells are pushed through a small narrow path, and in another path there was the droplets that contain basically the UMI and other barcodes,
The beads.
And they are pushed together into one single lane for them to undergo, I think, amplification or something. So my question here is, how can we make sure that one single type of cells are introduced to a specific barcodes, specific type of particular barcodes?
Okay, so basically you need to optimize the concentration of your cell suspension, or the rate at which the cells are coming from one inlet. So you have cells entering from one inlet, from one entrance, and beads coming from another inlet. So you need to have the right concentration. So it should not be too dense. Otherwise, you will have cells next to each other. So they should be dilute enough. The cells should be dilute enough so that they meet one barcode. So you need to optimize the cell concentration, the bead concentration, and the rate of the entrance of both. So once you have to repeat this experiment many times with different flow rates, with different cell concentrations or bead concentration. Then once you have your droplets, you need to check under the microscope, see whether you have mostly one cell in a droplet with a barcode. This is how it is done in the lab. And once it is optimized, then you stick to those concentrations and flow rates.
Okay, amazing. Okay, thank you so much.
You're welcome.
We have another questions from the audience. So the question is, again, related to the trajectory analysis. So how can you determine beforehand whether you need to do cycles or whether the linear trajectory is expected, whether you use literature and previously collected data to make this decision, or basically how do you proceed?
Yeah, that's a good question. I mean, in bioinformatic analysis, we always need to have some biological knowledge. So the bioinformatic analysis or trajectory inference, if I stick to today's topic, will help you to discover things that you don't know. But you need to start with something that you know. So you have a certain biological question, you should have a minimal knowledge. I think it should come from the literature or from pilot experiments, etc. And if you see that you have cell cycles, like if you have a cell culture, you definitely have cell cycles there, then you need to use the method that can detect the cell cycles. But if it is, I don't know, more of a differentiating system, like developmental, if you do developmental biology, then the question is more linear or bifurcation based. So is that you would use the methods that are more appropriate for that, or you just try the different methods and see whether they make biologically sense. This is how we would do if we have no idea.
Thank you very much. We have another question from the audience related to the Monocle, the MST3 path. So basically in your slides, Adam, you showed numbers from 1 to 9, and I believe it was not really clear what do these numbers actually mean and whether they are affected by the order of cells.
These numbers here.
Yes, I believe these are the numbers. Yeah.
So each dot is one cell, okay? And initially, the cells are in the high dimensional space, so you use dimensionality reduction methods, ICA or PCA, but if you use Monocle, it will use ICA. It will use the ICA to reduce the dimensions, and then you will get this kind of 2D plot. So the cells are basically projected in this 2D, and once you have that, you can measure the distances between each pair of cells, like this one and this one is 3. this one and this one is 6. The exact meaning of the distance, it doesn't mean anything actually. It's just to see that these two cells are closer than these two cells. Okay, it's just a value to see which cells are closer and not. Once you have distances between each pair of cells, then you can find the tree that has the minimum sum of distances. So if the position of one cell is changed, like this one is changed here, then of course the minimum spanning tree will also change because the distances will change.
Okay, thank you very much for the explanation. Let's jump back to the velocity topic. So the question is on the single cell velocity and the read depth. So if some genes have shallow sequences, will they work well with the single cell velocity, or how does the sequencing depth affect it?
No, it will not work well. Because first of all, this method works if you have good number of spliced and unspliced reads. And usually, so as I show also on this slide, you have much more spliced reads, like close to 80%, and the remaining 20% is unspliced. So basically, you need to sequence deep enough so that you also can capture the unspliced reads. Especially if it is a gene that is expressed, then you need to go even further in the sequencing for that particular gene. If you think that that gene might play a role in the description of the trajectory of the cell.
Maybe one last question from the audience that we got. So it's also related to the velocity. So if you have this one formula for the unspliced and spliced RNA, so does it always apply for all types of mRNAs? Like, can you also include the coding versus non-coding mRNAs? How does it work?
So this is a limitation of the method. First of all, it assumes that those parameters are not time dependent. And I believe that they can be time dependent, right? Because if a cell receives a signal, then its transcription on off will also change with time. So this is the first thing. The second is that the only parameter that is assumed to change with genes is alpha, the transcription rate. But beta seems to be independent of the gene and even set to 1. Okay, so, but there are other RNA velocity methods where they include the beta parameter, so not being 1, and also consider them as gene dependent as well. So then I would say, yes, we should consider these parameters depending on whether the RNA is coding or non-coding. But this is the limitation of the method, these assumptions. We also need to consider this into account.
All right. Thank you very much, Adam, for your talk and for answering all these questions. I hope that our attendees found it to be useful. The recording of this webinar will be available on our YouTube channel, probably within the next 2 days. So if you want to rewatch the session, you are very welcome to do so. And I will be happy to see you next time in 1 week to attend our last session of this bioinformatics series, this time with Stacy Zhu from the Singaporean office. So thank you very much and have a good day.
Thank you. See you.